Hello, this is Forrest Blocker, and I'm going to talk to you today about gene therapy. This is the second part in this series. In the first part of the series, we talked about gene therapy as it's being tried for some blood diseases and autoimmune diseases. This time we're going to be talking about all of the other diseases besides cancer that gene therapy is being tried on with a certain amount of success. I'm here with Maxwell Blocker.
Hi.
And let's begin by talking about how gene therapy can be used for heart disease. HGF plasmid DNA therapy of cardiomyocytes is being examined as a potential treatment for coronary artery disease and damage after myocardial infarction, in other words, a heart attack. There is a drug called neovasculogen. In 2011, neovasculogen was registered in Russia as the first-in-class gene therapy for treatment of peripheral artery disease. Neovasculogen is a plasmid encoding the CMV promoter and a VEGF peptide. There's another drug called Mitacar. Between 2007 and 2010, a phase 2 trial was conducted to test a therapy to increase the levels of SIRC-A2A protein in heart muscles, improving muscle function for cardiac patients using this drug. In December 2015, the cardiovascular gene therapy agent, Mitacar, did not meet its primary and secondary endpoints in the phase 2 CUPID-2 trial for systolic heart failure.
What's systolic heart failure?
Systolic heart failure is when the left ventricle can't squeeze enough blood into the body. The valve can't contract enough. Okay, the next set of diseases that I want to talk about are metabolic deficiency diseases. And the first of those is a lipoprotein lipase deficiency disease. Glybera is an adeno-associated viral vector expressing lipoprotein lipase for the rare lipoprotein lipase deficiency disease. In 2012, it was the first treatment approved for clinical use in either Europe or the United States. However, it failed to achieve market approval. The company subsequently decided to terminate further development of Glybera, saying refiling would just cost too much. Faced with the need to run 2 additional clinical trials to win approval of the product with an uncertain commercial prospect, Unicor has canned its plan to bring Glybera to market in the U.S. as well. So this particular drug cost a lot of money to develop. It did not have enough patients in the clinical trials to win approval, which was the primary reason that it didn't achieve market approval in Europe. So the company was asked to run 2 more clinical trials, and the company just decided it was going to be too expensive since the market size for this rare disease is so small. And this is not an unusual thing that we've seen in the history of development for gene therapy. The sort of the low-hanging fruit in gene therapy are diseases that are monogeneric, meaning that there's only one gene that's gone wrong. And for most of those diseases, they're rare, and so the market size is very small. So it takes years and years to develop the drug, and then the clinical trials, since there aren't very many people who have the disease, are very small, and it's very hard to win regulatory approval with very small trials. So this is something that the industry is going to have to figure out how to overcome if we're going to be able to make any progress with gene therapy. And as we saw in the last video, there are some encouraging gene therapy applications that would target a larger market for example, HIV infection. But those are more difficult to treat because in that case, we're trying to turn off a gene rather than introduce a missing piece of machinery into the cell from a single missing or mutated gene. So I'm not sure what the best way going forward for this, but clearly leaving gene therapy development in the hands of private industry is something that almost dooms it to failure unless there's just a spectacular success. And there have been a few very promising results. But again, if we expect companies to spend years and years and millions of dollars to develop a drug for a very, very small patient set, their stockholders aren't going to be happy. And that's the way that industry works. So what can we do as a society to encourage, if we want to encourage, the development of some of these therapies which might be, very helpful for people who otherwise would be likely to die? And should we do that? Gene therapy, of course, isn't the only possible technology to develop in order to help people with these rare diseases, but if we're able to develop this technology and eliminate the serious adverse events, particularly gene indels, which is short for insertion or deletion of the gene in the wrong place, then we can apply gene therapy to more complicated diseases that involve multiple genes, such as, for example, cancer. One of the ways that regulatory agencies do this is to give a drug a so-called orphan drug status. Are there other incentives that we can or should provide? And that's a question that is worthwhile to discuss. And in the later part of this video series, I'm going to talk more about some of the commercial and ethical implications of gene therapy, both of which have positive and negative aspects that have to be considered as we go forward. Okay, the next disease I want to talk about is adrenoleukodystrophy. It causes children to lose cognitive and motor skills. In 2009, researchers halted an adrenoleukodystrophy in 2 children using a lentivirus vector to deliver a functioning version of ABCD1, which is the gene that's mutated in the disorder. Bluebird Bio is also developing a lenti-D drug to treat this disorder.
What does ABCD1 do?
Good question. This is a protein that's involved in transporting long-chain fatty acids into an organelle called the peroxisome, where those fatty acids can be oxidized. Okay, so the next set of diseases is blindness, and there are a couple of different kinds of blindness that are caused by single genes that, for that reason, are appropriate targets for development of gene therapy early on. So gene therapies are being developed to treat several different kinds of inherited blindness. The eye is a convenient compartment for gene therapy because the retina is easy to access and it's partially protected from the immune system, and viruses can't move from the eye to other places in the body. There's a disease called Leber's congenital amaurosis, which is inherited and caused by mutations to the RPE65 gene. Delivery of recombinant adeno-associated virus, AAV, carrying the RPE65 gene yielded some positive results in 3 small independent clinical trials where patients recovered functional vision without apparent side effects, as reported in 2008. Similarly, 6 out of 9 patients with another disease called choroideremia had improved vision 6 months to 2 years after a virus was used to deliver a functional REP1 gene, which is the gene that's mutated in that particular. Okay, the next disease I want to talk about is Parkinson's disease. Patients with Parkinson's disease gradually lose cells in the brain that produce the signaling molecule dopamine, and eventually sufferers lose the ability to control their movements. A small group of patients with advanced Parkinson's disease were treated with a retroviral vector to introduce 3 genes to make dopamine. After treatment in 2011, all of the patients in the trial had improved muscle control. The next disease is hepatitis. Hepatitis B is a DNA virus that replicates by reverse transcription. The persistence of HBV, or hepatitis B infection, is due to the intrahepatic existence of HBV circular DNAs. In other words, the reason that HPV infection persists is that there are circular DNAs that stay in liver cells. It's hard to clear and serve as a reservoir for HBV reactivation. Early studies using antisense technology, ribozymes, and exogenous RNA that generate HBV RNA-containing artificial RNase P substrates failed to achieve efficacy in vivo. Okay, so those didn't work. The antiviral RNA I activator, ARC520, which contains cholesterol conjugated anti-HBV siRNAs and hepatocyte targeting peptide, was shown to be effective in primates, and early indications are that the drug is safe, well tolerated, and inhibits markers of HBV replication in humans. TALENs and CRISPR-Cas technology have been employed against the virus in the lab. So those are two of the programmable nucleases, which we're going to discuss in detail later in a later video. In Hep G2 cells expressing HBV in the lab, the introduction of circular DNA targeting CRISPR-Cas9 system results in both decreased hepatitis B core antigen expression as well as mutated circular DNAs. Okay, the next disease I want to talk about is cystic fibrosis. Using cultured primary adult intestinal stem cells derived from cystic fibrosis patients, the CFTR locus responsible for cystic fibrosis was corrected by homologous recombination, resulting in the clonal expansion of organoids harboring the desired genetic change. Data from 3 trials on cystic fibrosis transmembrane conductin regulator gene therapy, however, were not successful. Okay, so before I start discussing mitochondrial replacement, you'll notice that in this series there were several gene therapy drugs that were developed in the lab that showed promise by actually targeting and overcoming the gene deficiency. However, they failed in clinical trial, and they failed for a variety of reasons. Some of them introduced indels, which is an adverse event. Some of them did not deliver their payload to the cells, so they weren't effective. And some of them failed because the company couldn't support the research given the market size. All right, so the next one is controversial for another reason, which is an ethical reason, called mitochondrial replacement. For some children, if they're born with defective mitochondria, they lack the ability to make enough ATP. It can be a disease that is anywhere from moderate to very severe. So one of the ideas is, rather than introduce a gene, just introduce the entire mitochondria. From a targeting point of view, this is a much easier technology to perform. You're not trying to either temporarily or permanently add a gene. What you're doing is you're replacing a completely defective organelle, the mitochondria, which is the energy factory of the cell. So this has been done, and the way that it was done is to use a mitochondrial donor. So as you may know, the mitochondria is something that you get from your mother. The egg provides the genetic material from the mother, as well as the cytoplasm and all of the organelles that are going to be needed to be replicated for all of the cells in the body, including the mitochondria. What one can do is to take the egg and put someone else's, a different woman's mitochondria into that egg. So they've called these children three-parent children. But of course, in a way, they're not really, because the nuclear genetic material comes exclusively from the provider of the egg and the sperm, where the chromosomes are in the nucleus. However, mitochondria does carry its own DNA, and so you are introducing foreign DNA into that child, or DNA of a third person, by having a mitochondrial replacement. And as we'll see later when we're talking about the ethics of gene therapy, one of the places where most scientists draw the line is in doing something called germline therapy. That is to say, it's one thing to manipulate the genes of cells that aren't involved in reproduction so they don't get passed on to the next generation. But it's another thing to manipulate cells that are part of the germline. And we'll talk about why that is, but I think it should be somewhat obvious that this is a different thing, because you're actually changing the inheritable features of human beings. So clearly that's something that we should be very careful in doing, and in fact most scientists think we shouldn't do that at all, period. So this mitochondrial replacement is sort of intermediate between somatic cell genetic therapy, where you're just changing the genes of cells that are not part of the reproductive process, and germline therapy, where you are. Because in this case, those children who had mitochondrial replacement will pass that mitochondria from the third parent forever, through all future generations. The first germline therapy, if we want to call it a germline therapy, consisted of producing genetically engineered embryo in 1996, wherein faulty mitochondria was replaced by a working mitochondria from another woman. This resulted in a child named Emma Ott, who is now 17 years old and healthy. She was one of 17 IVF babies born after cytoplasmic transfer, which includes mitochondria. Tests on Emma failed to find any third-party mitochondrial DNA. Now that's an interesting finding, because if the third-party mitochondria was in fact successfully inserted into her cells, we would expect them to be replicating, and we could find at least two of the other children did have third-party mitochondrial DNA. So far, none of the children have been shown to have any adverse health effects as a result of these transfers, whether they took or not. The regulatory and ethical questions about this treatment continue to be debated fiercely, as they should be. Okay, so that's it for today. The next lecture is going to be about gene therapy as applied to cancer. Thank you very much, and have a look at the rest of my Farm Status webpage